happy that he's here today and talking to us about genomic networks, epistatics, and cancer. Thank you.
Okay, thank you, and thanks for the invitation to come and talk to you guys. I appreciate everybody coming out for the talk, and especially people that braved the cold to come across campus if you did that. So my talk today, I guess, is going to be kind of applied bioinformatics in the sense that there's not going to be a whole lot of bioinformatics per se in my talk. Instead, I'm going to talk about some this concept of epistasis and how we can apply it to how we can compute across large data sets, identify potential interactions between genes, and I'll define epistasis a little bit more formally in a moment, and how we can use it to identify new interactions among genes in cancer and how that relates to cancer progression and even potential clinical consequences. I wish I could remember the talk from the Conte Center because that was the interesting one. But this is the one you're getting today. So the first part of this presentation, I just want to acknowledge up front some of the people that have been involved. Audrey is a statistician in my laboratory. Xiao Wei is a wet lab person, and she started her own lab now in Beijing. Jason is a computational biology student, and Megan is a doctor. And these four people have worked together to do this work, to do quantitative mapping of genetic interactions in cancer. We've had a lot of help from our screening center staff. And I'm going to talk about the computational platform and some of the pipeline that we've developed in order to be able to do this work. So those of you who have gone to a few cancer talks have probably seen this kind of diagram put up before. This is Hanahan and Weinberg's most recent update on this idea that there are hallmarks of cancer. There are a variety of different phenotypes that are important during the onset and progression of cancer that range from genome instability and mutation to avoiding the immune system to sustaining proliferative signaling, et cetera, et cetera. And all these things work together in concert to go from the development of a cancer to the progression, ultimately to the metastatic disease that kills the patient. And one can think in terms of each of these different hallmarks, potentially incrementally adding to one another in a simple additive model where you have a set of genes that is important for cell proliferation, and that set of genes is different than another set of genes that's important for evading the immune system, and you sort of just add up all the effects. But we know that that's not true. We know that there's all sorts of interactions between and among genes, between and among pathways. And that a multitude of pathways in each one of these hallmarks are responsible ultimately for the phenotypes that are associated with that particular aspect of cancer biology. And that there are probably many interactions among the different biological processes that are going on during cancer progression. And so in classical terms, we can think about this non-additivity as epistasis, as Fisher defined it. And of course, epistasis, these are some old guys. Bateson initially described epistasis as a variant or allele at one locus that prevents a variant at another locus from manifesting its effects or masks its effects. That is, if I, for example, mutate gene A, or in this case he has gene B for black and G for gray. If I mutate one gene, then I get one phenotype. If I mutate the other gene, then I get a different phenotype. So masking, or epistasis in Batesian terms, is when you mutate both genes and you only get one of the two phenotypes. Whatever phenotype you get, the gene that that corresponds to in the single gene mutant is thought of as being genetically epistatic to the second gene. Okay? So this is how developmental biologists think about ordering pathways, and people who are converted biochemists think about ordering pathways in terms of genetics. And so this is one way to think about it. Fisher, on the other hand, was much more mathematical in his thinking about, well, about everything. And so he defined epistasis as a deviation from additivity of the effect of these alleles at different loci. So simply put, a phenotype, such as cancer progression, can be thought of as genes plus environment plus genes in environmental interaction. So that's sort of basic. And to Fisher, the genotype effect on the phenotype then would be, say, gene 1 plus gene 2 plus an interaction term between gene 1 and gene 2. And if that interaction term doesn't equal 0, then you have epistasis, or genetic interaction between the two genes. And so in order to, so for the rest of today, or the rest of this part of the talk, I'm going to focus on this Fisherian model, but we're going to come back to Bateson. Okay, so therefore you can imagine that you might have a genetic interaction term that's positive or negative that's happening among a network of genes, and it's this cumulative effect to a statistical geneticist that is a black box to everybody else, that then leads to the tumor genesis phenotype. And of course, cancer is even more complicated because it can be thought of genes, environment plus mutation plus interaction terms. And so this creates a huge matrix of interactions, possible interactions. So when I say mutation, you know that cancers are the result of somatic mutations, and therefore that's an important role. But of course, those somatic mutations can interact with the germline as well. Okay, so before I go further with the epistasis, I want to tell you how we and why we decided that we were going to get into this. So there's this challenge right now where there's a huge amount of data that's being produced on a whole variety of diseases, particularly in the cancer field. We have set up a cloud computing infrastructure at the University of Chicago, an open science data cloud with Bob Grossman, where there's over a petabyte of cancer data from the Cancer Genome Atlas and other sources. And so, you know, we need to be able to analyze that. We have one solution. It's going to be one of probably a network of solutions, we hope. And we've been able to use this BioNibus Protected Data Cloud, which is a trusted partner with the National Cancer Institute, holds all their data, downloads people's DBGAP credentials twice a day, and so forth. It's a nice resource for the community, and I encourage people to check it out. But we've been using it to do some of our own computations. And one of the things, well, let me just, you guys know about clouds probably. And you want me to go through it? Okay.
We're a member of the Open Cloud.
Yeah, so Brian and you guys are part of this. So the idea is, you know, you might have different medical research centers, you might have companies, individuals out there, academic centers. We have a cloud for public data, cloud for controlled genomic data. So this would be like the protected data cloud with the Cancer Genome Atlas data, where the NCI and the NIH control access to the data, and you have to have credentials, you have to have an IRB approval or whatever. That would be the controlled data. Public data would be like 1,000 genomes or ENCODE data. My lab's part of the ENCODE consortium group. We do a lot of ChIP-seq mapping. I'm not going to talk about a lot of it today, but we do a lot of it. And then we have a separate cloud that's like connected up to the medical record system. And so the key is to get all these things to, in a secure and compliant way, interoperate and talk to one another, and in a secure and compliant way to get the data out to the medical research community. And you know, the vision or the dream is that then you can, for example, if you're a biologist working on a project, send your sample to whatever sequencing core or what have you, or omics core, get some data back, upload it into the cloud into the appropriate place, and then leverage off all of the data, because you're not going to want to, I mean, most of you probably don't want to download a petabyte of Cancer Genome Atlas data if you to do a comparison of your 10 genomes to all of that data. And so you want to have a system where all of this is sitting, and you can basically just get some cycles to analyze your data in the context of all that other data. And so so we've got 1,000 genomes, we've got some NHLBI exome sequencing, we've got is ClinVar. I don't think he has Target yet, but this is pediatric sequencing. And I'm going to tell you briefly about 110 million EMRs that we've mined recently on this cloud. So one of the things that we've deployed on this, and we've also deployed on our local Cray supercomputing facility, is something we call SwiftSeq. This is a pipeline that uses, it's modular, it uses the best GATK practices for calling variants, for aligning sequences and so forth. And so when one does these kind of analyses, it's nice to have a unified set of calls of what the mutations are, or where the rare variants in the genome are, and so forth. And so we've worked with people in the Computation Institute, Ian Foster and Lorenzo Pesce, to set up this thing we call SwiftSeq, which basically optimizes. So this is like what you get out of the box with the Broad and et cetera software suites. And we've sort of optimized this to make the best use of RAM and CPU in order to reduce wall time and reduce our costs of operations as we're running hundreds of thousands of node hours on these machines. Okay, so now I'm going to bring us back into, you know, how we're using this resource to study genetic interactions. So we wanted to see what kind of computes we could do over these kind of data to make new discoveries. And one of the things that occurred to us is that because you've got all of these different complex pathways, complex interactions among genes happening during cancer progression, and we know that, but we don't really know what the matrix of those complex interactions might look like. We thought one way we might be able to use all this data is to ask the question, what mutations, what genetic alterations are happening together in the same patient, more in enough patients so that you're able to get statistical significance of this happening more than by chance. So if you have gene A and gene B, that they're co-occurring in the same patient multiple times in multiple patients, okay? And so we started with just a set of 536 tumor normal pairs, and we looked at copy number variants, and we screened by gene expression data as well. So if you have a loss of a copy, we'd also look for those gene expression levels went down. We also looked at somatic mutations, and we looked for significantly co-occurring among the top 10% of mutated genes. This identified 122 candidate genes. We pre-screened siRNAs because when you do siRNA screening, there's this so-called off-target effect where it's difficult sometimes to get the same phenotype with 2 different reagents or 3 different reagents against the same gene. And so we wanted to only use those genes where we have multiple siRNA reagents that were yielding the same phenotype and consistent quantitative phenotypes. And from that, we got 66 genes for interaction mapping. We used a robotic system that is directed by Sam Bettis, and who runs our core. It's one of the first cores that I set up when we started the institute. And we do basically automated high content imaging where we incubate the different siRNAs. We do individual, and then we do pairwise. So we'll do gene A, we'll knock it down, and then we'll take a bunch of phenotypic data, and then we knock down gene B and do the same. And we do that in replicate, at least 4 independent knockdowns for each siRNA, and we look at 10 different phenotypes. Then we do the double knockdown, and we do that in each combination as well. We do this high content imaging, and we do automated image analysis where the software draws boundaries around the cells and around the nuclei, and we can look for deviations in cell size and cell proliferation and nuclear shape and cell shape and so on. Here's some examples. Yes? Yeah. So here we're using a MCF10A diploid cell model. It's an epithelial breast cell. And so what we're then looking at are certain phenotypes that are it's not cancer, but it's phenotypes that are maybe relevant to cancer, like cell proliferation. So there we count how many cells. So we knock something down. You don't think cell proliferation is relevant to cancer? Okay. Well, most of the cancers that we're treating at the University of Chicago have out-of-control cell proliferation. And so this is something we thought was relevant. Okay, that's true. And if you add MIC, like it'll even go faster. And then you can use them in xenografts, but that's a different talk. Yeah. Well, just development and progression. I mean, what we're looking at right now is going to be whether genes that we predict to be interacting, okay, using the statistical method I just outlined, to be significantly identified in the Cancer Genome Atlas data as co-occurring, whether those that we predict might be interacting are actually, in fact, interacting when we use a cell model. So we actually could have used a neuronal stem cell as our model and still test the basic premise of whether they genetically interact. What we chose to do was to use a breast epithelial cell that was diploid or largely diploid, and to assay a handful of characteristics that we think are related to or relevant to cancer phenotypes, like cell proliferation or failure to apoptose upon cues, or maybe cell shape, certainly cell size, can be attributes that are also observed in primary tumors. Are we going to claim that this is a great model to understand the tumor progression? No, that's not my point. This is a talk about the genetics of gene interactions and how one can go from looking at statistical patterns in data using bioinformatic approaches to actually showing that those statistical patterns are relevant biologically. So that's what I'll try to convince you of here. So we have lots of different phenotypes. And these are just correlations among them. And so this is more or less the kind of model that we're employing. This is sort of a simplified version where you can imagine that if you have a mutation in gene B, you might get 80% of the wild type phenotype. If you have a mutation in gene A, you might get 60%. In a multiplicative model like this, and simply put, 0.6 times 0.8 is 0.48. And so if you have a deviation from this 0.48 that's significant, then we're calling that a genetic interaction. And then within a confidence interval that one can measure, since we've done lots of replicates, we're able to actually give a quantitative estimate of what that interaction term is. So that's the sort of thing that we did. And surprisingly, actually, or a little bit surprisingly, of the 1,457 gene pairs that we tested in this matrix, of which we did about 20,000 different independent experiments to generate this data set, we found that there are about 850 significant interactions. And we distilled these down, we collapsed these into 3 major phenotypes that were not correlated with other phenotypes. So of the 10 phenotypes that we looked at, these are the 3 phenotypes that were representative of the 3 main classes that were not correlated with one another heavily. And you can see, cell number, cell proliferation is probably the biggest one. There's structure in this data when we actually cluster the genetic interaction terms that we quantified. And that structure, at least to some extent, may reflect interactions that at the molecular level may be happening. Like here's several of these genes are involved in DNA damage checkpoints. Here's TSC, PI3 kinase signaling are also related pathways. So one of the things that really struck me about this, and I thought was unexpected, was back to the Batesian view of epistasis, where you have complete masking, or you could call it complete dominance or masking. The majority of these genetic interactions are, in fact, showing this masking, this sort of Batesian style epistasis. And I thought this was surprising simply because we're looking at simulated hypomorphs where we're knocking these genes down, and we're only, we're not having nulls here. And so, yeah, this is what the data is showing us. We also saw considerable amount of synthetic enhancement or synthetic suppression. When we start to dig into what are the genetic interactions, what genes are they between, what do they encode, we see all sorts of examples like protein-protein interactions, kinase signaling, competitive binding at substrates, and transcription factor targets. I'll show you an example or two of little networks that we can build of transcription factors and their targets that are genetically interacting. This is one of them, where GATA3, we had previously published a paper where we showed the targets of GATA3 among other targets throughout the genome. And we see that GATA3 is an interaction network with these 3 genes, PPAR alpha, patch, and NF1B. And so they're both interacting at the level, if you looked at gene expression correlations, and it's a genetic interaction. And they're direct targets of GATA3. Here we have an example of 2 different members of 2 different pathways, a MAP kinase signaling pathway and a DNA damage and repair pathway. We hypothesize that perhaps these are all, so they're all interacting within and they're interacting between the 2 pathways. And we hypothesize that these may be due to commonality at both are known to target gene kinase signaling. So, however, when we sort of try to add everything up and we look at all the annotated interactions, all the inter-pathway interactions supported by the literature I just mentioned, even ones where we sort of expand it out a little bit more and we say, are you in a network that's interacting with someone else, with something that's interacting with another network? The vast majority of the genetic interactions that we're seeing in these among these predicted tumor genes is unknown. They were previously undescribed. So I think that gives us a sense of the complexity that is still yet to discover out there, the biological complexity that we have to unravel. This is really the first view I know of. There are people who have anyone's gone in and quantified a genetic interaction matrix like this in a cancer or based on cancer data. And probably the most, to me, potentially impactful and surprising thing was that we see that this data is also associated with predicting patient survival. And so in this case, one can have any of these three factors that are interacting with one another, and you get a worse outcome. In this case, we have two different genes. You can mutate one, and you don't see a significant deviation in survival from if you have neither mutation. You can mutate the other, and you see the same effect. But when you mutate them both together, then you have a precipitous drop in the survival of these patients. Many of these are also potentially druggable targets. And so, to me, what this is saying, and what I hope we can do in the future with these data, are help to guide us both in looking at how genetic interactions might lead us to making sensible choices about what kind of therapeutics to either try in preclinical or even clinical settings. And also by understanding the architecture of what interacts with who. And if you inhibit one, do you need to inhibit something else in a parallel pathway? Or are you wasting your time because, in this case, maybe you could do either one. And so sort of just thinking generally about the therapeutic implications is something that is starting to emerge in our minds because of this surprising finding that, in fact, these appear to be clinically relevant. I'm not just pulling these out as anecdotes. When we look at all this entire set of interactions, we have gene pairs with different outcome when co-altered versus gene pairs with genetic interactions. And what we see is there is a significant, it's still a small sample size, and we need to expand to the entire TCGA atlas, and we want to do more of this kind of thing. But it's looking like it may be significant when we look across all the data. That so just to recap, if we see in the primary tumor genome sequence data significant co-occurrences, and then we're able to validate those co-occurrences in a cell-based model, and then we go and we look at the consequences in terms of does that correlate with patient outcome. We're seeing an enrichment in our ability to predict patient outcome by taking this kind of approach. Okay, so I'm going to tell you one more story about Epistasis in a little bit of a roundabout way, and then I'm going to end with some work that we actually have done to do combinatoric mapping originally in fruit flies, and ultimately has led us to understand something we think more about kidney cancer. So let me start with this little vignette here, or this detour. So one of the other things we've done with this cloud-based system is we've been trying to pull in medical records. Andrei Rajetsky happens to have a set of 110 million different patient records where he has ICD-9 and 10 codes. Most of those are coming from the United States. We also have entire country of Denmark. We have a number of different systems that he's mined the data from. Texas, San Francisco, University of Chicago, and so forth. And what this is showing is what Andre did is he asked what are the comorbidities between different ICD-9 code diagnoses for both complex traits and Mendelian traits. So that is, if we look at 110 million people, when do we see, so much like I just told you about the co-occurrences, significant co-occurrences of genetic mutations in cancer, here we're looking at significant co-occurrences of traits that are happening in people that have been diagnosed with different diseases. So we can look at correlation between actually Mendelian diseases, and there's a surprising amount of signal in there when you look at 110 million patients. And we can look at Mendelian by complex, or you can look at complex by complex. And what this is just showing is that the log-odd ratios the distributions are highly correlated among all of these pairwise combinations. So it's not geographically constrained. It's like these are different hospital systems in some cases even different billing code systems that have been normalized so that you can compare them to one another. And they're even different countries, as I said. And so the data are very consistent from site to site. Yeah.
What is the difference between co-occurrence or comorbidity and being part of the phenotype but being a different ICD-10 code? So what I mean is, it's of course very interesting if you find a correlation.
Oh, disambiguation of, yeah.
Well, fine, because that's a very interesting thing that is being calculated.
Yeah.
But then there are certain things like alcoholism and depression where the psychiatrist will say, well, this is really part of the, alcoholism will often induce depression will induce alcoholism. It's really mechanistically related, and it's a comorbidity that comes from the primary disease,
right.
And it's just another symptom of that disease. And of course, with the mitochondria, how are you taking them out?
We're not.
Okay. So some of these are the kind of things that every physician could have told you.
Some of them are. Okay. Yeah. And in fact, those are things that are clearly in the data. We're looking at such a large number of diseases and so what we're looking for are the sort of the surprising things. But yeah, so that particular issue we've discussed a lot, as well as a lot of other issues, disambiguating different aspects of this data. But that is something that we're not making an attempt to actually do that right. We really have to dig in and manually go through with a team of 1,000 physicians, probably, all the data to try to find the things that maybe, and we still wouldn't be successful because the mechanistic connections among all these things are not known, but some are, and those are in a sense positive controls. In this case, yeah, they are diseases, correct? Or sorry, just let me correct that. They are the log odds ratio of the probability that these two are significantly associated. Two diseases. Yeah. Right. Okay. Sorry, you had a. Yeah. So some people will have multiple diagnoses, and that's actually what gives us the power to do this analysis, because it's multiple diagnoses in one person. Yeah. Oh, sorry. So that individual. So let's say we had an individual with 3, then they would have A and B, and A and C and B and C. Those would all be co-occurring. So okay. So this is just showing that Mendelian diseases co-occur significantly. You can see here's an example of urea cycle metabolism and thrombocytopenic purpura having a high significance. So we see a lot of this kind of structure. One can cluster that, and one can find both cases of non-surprising and also surprising interactions. But what I actually wanted to focus on, this is published recently in Cell, by the way, so you can go if you're interested in going into the depth into this paper. What I want to just cover from this paper is actually these associations between Mendelian phenotypes and non-Mendelian phenotypes. And the concept here, and I think Andre just had a brilliant idea here, it's very simple, is that for the Mendelian phenotypes, we often know the underlying genetic mutation. And so basically by the transitive property, if you have a correlation, if you have a comorbidity between a complex disease and a Mendelian disease, then perhaps that gene is also involved somehow in the complex disease, right? So by applying that concept, we were able to put together what we've been calling a Mendelian code for complex disease. That is, each and every complex disease has a series of Mendelian diseases that are comorbid with it to a significant level. So if someone is diagnosed with a Mendelian disease, then they're more likely to be diagnosed with some corresponding complex disease. And so one can cluster that, one can find, again, some, I don't know, some people may or may not find this surprising, but look, you got a very high bootstrap value between bipolar disorder and glaucoma, for example. So I thought that was surprising, and so on. So how do we test this? So we sort of thought about this for a while, and I eventually decided that probably the biggest data set we could look at is by looking at all these GWAS studies that have been done for all these complex diseases, and then ask, do Mendelian code genes, are they enriched in their representation as GWAS hits? And the answer is yes, they're highly significantly enriched. And then I asked Dave Blair, who's a graduate student who did all these computations with us, if these are more likely to replicate in GWAS. That is, if you have two independent GWAS populations that have been measured for the same trait, say breast cancer or something, then the sites, the SNPs that are associated with genes that are replicating between the studies are thought to be the ones with the best signal, the most reliability. In fact, that's when it really gets strong. These Mendelian code genes are much more likely to replicate in this study. So what we want to do next is actually take these data and predict genetic interactions, considering these as genetic interaction predictions, and then try to find cell models like we did in the earlier example to sort of knock down one and then the other and then both and see if we actually see genetic interactions. We haven't started that yet, but what Andre has done is he compared an additive model a la Neil Risch that people are typically using for the last 20 years or so, and he modified this with the concept of communities to define epistasis as interactions between genes that are in different communities or modules or pathways. The idea here being you need a hit in at least one gene in a module or in a community, and I can talk later about how we might define communities or how he defined communities here, but and then also in another community. And if you have both of those, and that interaction term between those two is in this alternative model here. And then we ask, do the data better fit the original additive model or this modified model that has this interaction? In fact, the data better fit the epistatic model. And so just keeping with the theme here of the talk, epistasis, this is another line of evidence that epistasis may be very much widespread in human disease. And so only available because we're the number of individuals that were looked at in this case. Okay, how much time do I have? 10 minutes? Okay, I'm going to go really quickly through the last part. And here's it also, not overtly has anything to do with epistasis, but certainly genetic interactions and networks of interactions among genes and proteins is a major part of what I'm going to talk about here. And I'm actually going to start with the Drosophila genome. We've been mapping the regulatory elements in both the human genome and the Drosophila genome for the last 5 years or so as part of the ENCODE and ModENCODE projects. You guys probably all know about ChIP-seq, and half of you probably have done analysis on ChIP-seq, and we've mapped all sorts of chromatin marks, and we've mapped throughout time, and we've mapped all sorts of insulator proteins. These are insulator proteins or proteins that create boundaries between genes so that regulatory element of one gene doesn't activate or control the expression of a regulatory element in the next gene over. We've mapped more than 100 site-specific transcription factors, and we're slated to map several hundred more and up to 1,000 in a project that we're doing with Mike Snyder at Stanford for ENCODE. Okay, so we've done all this mapping. We can cluster these binding sites, and we see lots of interactions. We can build networks, also seeing regulatory interactions among many factors, and we can start to use this data to make predictions about how sets of genes, which sets of genes might be regulated by which sets of factors in the genome. We've gotten fairly good in this model system of actually, figuring out precisely which transcription factors over development might be explaining the regulation of specific genes. So like the theoretical predicted is in dotted here, dotted line, and the actual observed is the straight line or the solid line. And so for many genes, we're actually able to do a really good job of predicting using sort of standard regression and machine learning approaches with these kind of data. So what we'd like to do is take these data and also make predictions about networks and how factors in complex networks are contributing to complex phenotypes. And so, about 70% of the genes in the Drosophila genome are actually conserved in human. And many of the genes that are involved in early embryogenesis are actually important in human cancers. And so what we decided to do is study some genes in the segmentation regulatory network, which has been heavily studied. Eric Wishaus and Nuslein Bohar got the Nobel Prize for their amazing early work in identifying these genes precisely because it turned out that these genes have orthologs in humans, and those orthologs are involved in a wide range of human diseases, and many of them, such as these segment polarity genes, are involved in cancers. These are all transcription factors, all these maternal genes, gap genes, parallel genes. They're called gap genes because when they're mutated, you lose gaps in the embryo. They're called parallel genes because you lose every other parasegment in the embryo. And the targets of these genes, as I just said, are often pathways that are important in cancer, such as the Wnt pathway, the hedgehog pathway, the TGF-beta signaling pathway, and so forth. So just looking at a network around a subset of this data is where we decided to start. And we decided to look at the FUTS and Eve homeodomain proteins. These are transcriptional factors that are expressed in this beautiful pattern, this stripe pattern, that are responsible for setting up the segmental enlage of the animal. We can map the direct targets by doing CHIP, as well as gene expression profiling in the mutants and seeing which of the CHIP targets are affected in the mutants, and that gives us putative direct targets of FUTS and Eve. And we can build networks by using these kind of data and combining these kind of data together, and combining these kind of data with things like protein-protein interaction data. And this is an old paper that Andre and I had with Ivani Yosifov, where we look at combining noisy data from heterogeneous data sources. And so this is sort of roughly the kind of approach we use to build our networks, where you can build joint probabilities, and then you can lay down a network at random, and then you can compute across these joint probabilities, each of these probabilities would be like the probability of two proteins interacting, or which has its own model, or that a gene is differentially expressed, which has its own model, or a gene being a target of a transcription factor, which in turn has its own model. So it's all sort of layered in. And then what you do is you change the network after computing this joint probability, and you remove an edge, or you move an edge, or you remove a node, and then you recompute it. And you do this millions and millions and millions of times. And you through Monte Carlo sampling method, and you then try to calculate, or you in fact do calculate a probability for each type of interaction in the network, and you get a big hairball. But the hairball has, each edge has a probability associated with it that is weighted based on these type of input information. And so what we want to do is explore this network to see if we can find anything new about these pathways that were aforementioned. And one of the things that struck us was this particular gene, CG9924. And the reason it was really interesting to us was threefold really. First, it has the highest betweenness centrality in the network. So to get from any one point to any other point in the network, you have to pass through this gene the most frequently. The second thing is that it interacted with some interesting factors. In fact, this Jun kinase signaling pathway, the Jun kinase phosphatase puckered interacts with it. And we thought that was interesting because this pathway is important for cell morphogenesis in these animals. And that's an important part that there's little understood in terms of how the segments actually form biophysically. And so we thought maybe this was targeting some interesting things. It's a direct target of FUTs, direct target of E. And the third thing is that it's highly conserved between Drosophila and human. It has math, BTB domains, and is predicted to be a member of a Col3 ligase complex. And at the time, there was just a couple of papers. So this is almost a decade ago when we first found this thing. It's called SPOP. And it turns out SPOP, fast forward today, has gotten really hot because it's mutated in prostate cancers, it's mutated in other kind of cancers. And I'm going to tell you what we've found in a cancer it's not mutated in, but it's doing something quite different. You know, it's expressed in stripes in the embryo, and the stripes go missing in the mutants. It localizes to the segmental grooves where these morphogenetic movements are happening. Another group calls it roadkill because it has this phenotype as during embryogenesis. It does have lons identicals and some of the classic phenotypes from the Nusselt-Volhard screen. They probably missed it for two reasons. One, it's just messed up and it doesn't give a clean phenotype. And two, it has a strong maternal component. And so I can get into that later why that's important. But the point is that this is not a very good phenotype to look at. So we turn to the fly eye, where we activate the TNF signaling pathway through Jun kinase. And we can either activate it weakly or we can activate it with a strong allele. If we activate it with a strong allele, it causes cell death. Here it's just on the edge. And so what we want to do is we want to put it in take this sensitized background, and we want to ask, what effect does putting an SPOP mutation have on this? If the effect is to activate puckered, then it would have a negative effect on Jun kinase, and you would lose some of the eye here. If it's inhibiting puckered, then the eye should come back. The eye comes back, and this makes sense in the context of this being a potential puckered being a potential target of SPOP as part of the E3 ligase complex. And so we tested that. We show that puckered is indeed reduced when you add SPOP at the protein level. It comes in these two isoforms. And when we block degradation, we get hyperubiquitalization of puckered. And we show that, I'm not going to go into the details, but we show that's true in human as well. But the sort of, how does this relate to cancer? Well, many of the genes that are identified in this pathway, I'll say once again, have been implicated in human cancers. And so what we did is we thought, well, here's this highly conserved thing. It's conserved in Jun kinase signaling. It could have other targets too, and I'll show you that it does. but is it involved, is its ortholog involved in human cancer? So what we did is we just asked, is its expression level altered in human cancers? And so we looked at 20 different tumor types, and what we discovered was that in clear cell renal cell carcinoma, almost 100%, basically 100% of the clear cell renal cell carcinomas are showing high levels of staining. And in normal tissue, they're almost always negative. The other thing that is notable about this is that it's mislocalized in the clear cell renal cell carcinomas. So this is a CACI2 cell line, which is derived from clear cell renal cell carcinoma. You can see it's in the cytoplasm, whereas in a HEC293 cell, it's in the nucleus, and other cell types, it's in the nucleus. So we're only seeing it in the cytoplasm of these cancer cells, and in the primary tumors, there's cytoplasmic localization as well. So we wondered if that was important. So what we did is we took the nuclear localization signal off of it. It does have a nuclear localization signal. It's 6 amino acids at the C-terminus. We lopped that off, and then we put that back into HEC293 cells, and we show that that causes hyperproliferation of these cells, whereas we had shown before that SPOP, and this is actually now accepted to cancer cell, this will activate the apoptotic pathway with the full length. So the full length is going to the nucleus. This version is going to the cytoplasm. When we knock down SPOP in HeLa cells, we don't see additional cell death, but when we knock it down in the clear cell renal cell carcinoma cell line, we see increased cell death. So at least in cell lines, it looks like it's both sufficient and necessary for proliferative phenotypes. So then what we have done is established both subcutaneous and subcapsular, where we do operations on these mice, and we inject it in the subcapsular region of the kidney, and you get these very big and heavily vascularized tumors. When we do this, we don't get it with SPOP, of course. We also don't get it with the control. So it's only when SPOP is in the cytoplasm that we drive these tumors. So what's going on? In kidney cancer, we know that HIF is involved, hypoxia-inducible factor. VHL is frequently mutated, the most frequently mutated. It's at least 70–80% of kidney cancers have VHL mutated in it. And VHL actually targets HIF for ubiquitination, and that happens in the cytoplasm. So here, VHL goes to the nucleus, it targets things, and that causes cancer. Or HIF goes to the nucleus, targets things, and that causes cancer. We show here that SPOP actually accumulates in the cytoplasm under hypoxia. These are different cytoplasmic or normoxic fractions. And then you also see this in cell lines, although you have to turn the lights down a little more to see that. SPOP is directly regulated by HIFs, so we mapped HIFs genome-wide just to find binding sites in SPOP. We took this binding site, cloned it out, showed that it's actually has HIF-responsive elements. We mutate those elements, and it's no longer HIF-responsive. We can overexpress HIF, and it overexpresses from that element. We can knock down HIF, and it underexpresses. We got the crystal structure with puckered, and we've got crystal structures with some of the other targets now as well. And we identify this domain here that seems to be binding all of its substrates. And we wanted to know, does it have some new substrates in the cytoplasm when it goes off to the cytoplasm? And so what we did is we took this, what we call the substrate binding consensus, and we looked and we asked whether this degenerate sequence is found in proteins in the genome. And we're particularly interested in phosphatases because puckered is a phosphatase, and so we found USP6 and 7, which are ERK kinase pathway members, as well as PTEN. And the crystallography was done with Brenda Schulman's group, who was very interested in this because it was a first in class when we first found it. And this is just a bunch of figures that are showing you, and because time is short, I'm not going to go through them all, but it's showing you that PTEN, in fact, is a direct target. It gets ubiquitinated as a result of interacting with SPOP, and we can do pulse chase experiments and all those sorts of things to show that both in vitro and in primary tumors, you can see when PTEN is down, SPOP is up. In normal kidney, P10 is up when SPOP is down. The USP is also similarly regulated, and we can knock down USP6 and 7 together, and we get hyperproliferation of these cells. We show the same thing for GLI2, which is a pathway, another target in the hedgehog pathway. It's a transcription factor in the pathway, the hedgehog pathway, and DAX, which is a pro-apoptotic molecule. So all these things are targets of SPOP. It's acting as a hub, just like it was the hubbiest, most central factor from that Drosophila network. It's also acting as a hub in kidney cancer, and it's doing it in an artificial way when it gets mislocalized to the cytoplasm. Now, artificial in the sense that normal, it's causing cancer. Now, it may in some other cell types or some other time get localized to the cytoplasm in humans for a good reason, but we don't know what that reason is yet. Okay, so just to finish, we identified through this process that in normal cells, SPOP is in the nucleus expressed at low levels, but when VHL is mutated, HIF goes to the cytoplasm. It targets SPOP. It also targets VEGF, and VEGF leads to additional vascularization and angiogenesis, and so you supply the tumor with more blood. It grows. SPOP is making it proliferate by this novel mechanism of downregulating P10, as well as several other substrates and other important pathways for cancer proliferation or blocking apoptosis. And so you can imagine this is sort of like a really nasty loop. So then the tumor grows, you get more hypoxia, more HIF, more SPOP, more VEGF, more tumor. And so this is, and what we're working on right now is we're trying to see if we can't develop small molecules and humanize antibodies against this domain, the substrate binding consensus domain, to see if we could specifically inhibit SPOP's ability to interact with targets. So, so that's it. And I hope I convince you that there's a heck of a lot of genetic epistasis happening in the human genome, in human cells during complex disease like cancer, that we can use these kind of methodologies to identify new interactions that are predictive for patient outcome. We can also use these kind of genetic network interactions and other kinds of data to build up networks that are predictive of which molecules might be important as substrates or as drivers of complex biological processes, and then we can dig in and determine whether or not indeed they are important. And so the people in green actually for this talk, they're on the ENCODE project and on all of our nuclear receptor stuff, so I didn't talk about that. But Subhadeep, Jian, they really led this this work on the SPOP story. I showed you pictures of everybody else, so thank you.
Thank you so much for the talk, and wait for the microphone to come before you ask the question. Hi.
Right over here, Al. While we're waiting, Kevin and Smith and I and John Ianian at IHPI are working a contract with Optum Labs and Optum Insights so we can have access to about 110 million records. And so we'll be calling a meeting soon to convene folks to see what you want to do with the data from those resources, because that's a fantastic thing that's going on there. A lot of cool stuff there.
Yeah, there's a lot of directions you can go with that data.
Yeah, so that's really inspiring. Go ahead.
I have a very quick question relating to the comparison of the Mendelian diseases and the common complex diseases when you looked for the SNPs that actually are deleterious and if they replicate in the GWAS sets. Because for most Mendelian diseases, the SNPs are in coding regions and the majority of the GWAS sets are in non-coding regions. So you probably lose a lot of information right there.
Yeah, that's a great question. And we've been having this internal debate Andre and Nancy and Cox, Nancy Cox, and myself. You know, what should we do? Should we go and look for rare variants associated with these gene lists and do a bunch of sequencing, case controls? Should we take this genetic interaction approach? What should we do to try to further validate and figure out, you know, what are the important constituents from those lists? So far, what we've done is we've looked at exome data that's available from a couple of complex traits, and we can do burden tests and that sort of thing, and we get nothing. So consistent with the rest of the field. And so that's where we are with that, and we're about to actually get, I think, 10,000. They're either schizophrenia or bipolar that have been done, and maybe that'll give us more power, and we'll be able, because we haven't looked at that big a number yet. But you know, your question is about the non-coding, and so I think that's a great idea. And we've actually got this assay lined up where we can do high throughput functionalization of enhancers, where we're capturing these putative enhancers. We're calling them downstream of minimal promoter, and then we transfect all that back in at once, and then we can just it causes its own transcription. And I have another talk on that I can give you. And then we just do RNA-seq, and we can quantify it comes off. And so Alex Stark in Austria has published something similar. And so in flies, in fruit flies. And so so everything, that's the other message I want to get across to all the students here. Everything, you know, model organism research is really important, because almost everything that we learn in biology has started with model organism research. And so often people, you know, develop these techniques, and then I could give you a talk on the human stuff, and I just talk about the human stuff. And yeah, I do this in humans, but it's the fly guys that like figured out how to do it. So, or the yeast guys. And so just keep that in mind. I think that combined with deep sequencing to see, because it could be common it could be more common polymorphisms. It doesn't have to necessarily be rare alleles. So it could be common polymorphisms in these regulatory regions that are contributing. So I think that's a great idea, and that's definitely one of the directions where we want to go with it. Yeah, yeah. Well, we can use that monogenic gene list to find, along with the GWAS studies, to find where those SNPs are. And then we can overlap that with all the ENCODE data we've been producing and in that project, and ask where are the potential enhancers. And that's exactly, I mean, we have a project to exactly do that right now, that we're starting to use this methodology to pull it out. Whether we want to do deep sequencing or not, I don't know. I think we can just, well, right now we can start with the common variation and see what can we learn from the common variation. How much is that actually affecting the phenotypes? And you can add more layers with the rare variation. You say, well, do you find rare variations in binding sites where we have position weight matrices where we can predict that it might change the energetics of the interaction between the transcription factor and the piece of DNA and all that sort of thing. So there's cool stuff you can do with that idea.
Yeah. So, Thomas.
Do MCF10 cells count as a model organism or as Homo sapiens? As a statistician, I'm interested in your comparison of Bateson and Fisher, and you came back to it, but much too briefly in the middle of the talk. For the, if I'm right, 1547 gene pairs, yeah, where short hairpin knockdown produces an apparent interaction. Can you say a little more what the range of phenotypes for single knockdowns and the pair knockdown are, and in what sense is it that you find complete suppression of the phenotype rather than harsh, a Fisherian quantitative epistatic interaction.
Right. Well, yeah. So I think maybe part of your point here is that you might hit a threshold where a cell has become only so oblong, right? And so if you have a little bit oblong and maximum oblong, a little bit plus maximum is still maximum. In which case you would show masking or classic Batesian. So I'm pointing out the caveat to this, but we see a whole range of phenotypes, and it's quantitative in the sense that we're measuring certain parameters such as total volume in the cell, or not volume, but in 2 dimensions, but and so or we're measuring how many cells are in a given area, so how much proliferation, so we're just counting, and so we're saying, okay, well, we've got 300 plus or minus 6, and so it really is it really is something that is a range of phenotypes that can be quite broad. And so it's both the phenotypes are broad and the measurements that we're making are fairly precise, and you can have something that's very misshapen, you can have something that's slightly misshapen, something that's proliferating very strongly, you can have something that's just barely detectable. So it's quite a dynamic range, I think, and I could.
And so you're
what you'd
suggesting,
probably prefer is if I actually could show you a chart of the dynamic range and tell you it's over. But I would say it's sort of like maybe over an order of magnitude or 2.
But what you're suggesting is that the appearance of masking is due to limitations on the phenotype.
No, I'm saying that that's a caveat.
Thank you.
Yeah, but that.
And so you do see apparent masking even when there's not a technical limitation on the phenotype.
Correct. Yeah. No, I would say that's the majority of what we see. Yeah, I don't think we're at those maximum thresholds very often. Yeah.
So at the very beginning, you mentioned an interaction that I'm very interested in, but that didn't come up again, which is the gene-environment interaction. And if you have co-occurrence between Mendelian diseases and complex diseases that have apparently nothing to do with each other and nobody thought about, one possible explanation could be a common environmental toxin or an environmental factor that influences both your glaucoma and your bipolar disease or whatever.
Correct.
Is there any way you can distinguish between gene-gene interaction and gene-environment interaction that actually triggers this co-occurrence? And of course, in the translational thinking, that is maybe even more interesting because we can't change our genes, but we can change our environment.
There is a way to do that, and it's with deeper data, and so it's hard to capture whether someone's a smoker from their billing code.
It's gonna be
Well,
easier now.
EMRs?
Maybe you, okay.
Easier now.
That's not in those.
It is. There's laws now with the meaningful use that has to be coded, so that could be pulled down.
Okay, well, all right. So that was a wrong
It's
example.
not buried in the notes anymore.
Yeah, but
Yeah.
I think whether you're vegetarian or not, I mean, the BMI will probably be in there as a
But,
smoking, but, whether you're vegetarian or not, or whether you're eating a lot of
There's
junk
all sorts
food,
of
that
there's
kind
all
of stuff
sorts of
is
things
usually
that-
not
that's
in
not in the
medical
billing
records.
code. So having deeper data, and maybe it's going to be hard with the Mendelian associations. I think it'll be less hard with the complex associations. So that's probably where you would start. So you look for complex by complex, and then try to factor the environment in to it. So we've been thinking about various things like, can we overlay? Is there any way we can get? We're probably not allowed to do this, but deconvolute the patient's geography, where they're from, from their records.
To do that. The zip code that you would.
Yeah, so,
It's somewhat controversial.
yeah, so,
Like one of the 19 or something.
yeah, so,
Oh, it's definitely, it's one of the,
Yeah, so-
it's right in there with your name and PID.
yeah, so there,
Moira knows.
but one can, one can imagine that you could use that kind of data as a proxy for environment. And Andre's actually been doing a bit of that.
There's a lot of that work being done here. Brandon School of Natural Resources and Environment came to do called geospatial health.
Yeah. And then we're even getting crazier sometimes in how we're thinking about this, because like Grossman has his own satellite with NASA, and he's taking like real data, real-time data. It's true.
We work a lot with him.
And it's called the Matsu project, and they're like taking Earth, longitudinal, spectra of the Earth over time, and they're generating terabytes of data all the time, every day. Yeah, it's danger. He can zap you from outer space.
This is a great note to thank you again for a really very interesting talk, going anywhere from environment and genes and base and Drosophila to outer space. So thanks for coming by.
All right, thank you guys.